Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on genetics. The last time we were together, we learned about the molecular structure of DNA and its 4 types of nucleotides. Today, we are going to discuss DNA replication. Ready to get started? The ability of a DNA molecule to self-replicate is very important. In fact, it is the basis of all methods of reproduction, as well as the basis for passing on genetic information from one generation to the next. When a cell divides, it is critical that the daughter cells contain the same genetic information as the parent cell that produced them. Students, there is one exception to this rule. During what normal process do dividing cells not produce exact replicas of themselves? Take a few minutes to think of the answer to this question.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I hope that the answer to this question was obvious. Cells do not produce exact replicas of themselves when they are forming sex cells. It is important to note that this is the only normal circumstance in which this is the case. Good work. DNA molecules exist within chromosomes in the nucleus and are surrounded by a sort of soup made up of free DNA nucleotides. These are the components that will be used to build new DNA molecules. DNA replicates semi-conservatively, which means that each new DNA molecule formed contains one strand from the original DNA, and both new DNA molecules formed are identical to the original molecule. Now, let us take a look at the process of replication. The process begins at particular points in the DNA, which are known as origins. These are targeted by proteins that separate the two strands and initiate DNA synthesis. Origins contain DNA sequences recognized by replication initiator proteins, which recruit other proteins to separate the two strands and make replication forks. Origins tend to be rich in adenine and thymine bases, AT-rich, because AT base pairs have two hydrogen bonds, rather than the three formed by CG pairs. Therefore, AT strands are generally easier to separate because they are attached by a smaller number of bonds. DNA helicase enzymes break the hydrogen bonds and unwind part of the helix of the DNA molecule. This reveals two single-stranded regions. Once the strands are separated, RNA primers are created on the template strands. Molecules of DNA polymerase follow the helicase along the single-stranded region, which acts as a template for the synthesis of a new strand. The DNA polymerase then assembles free DNA nucleotides into a new strand, alongside each of the template strands. The base sequence in the new strands is complementary to the template strand. Why? Because of the base pairing rule, adenine can only be paired with thymine and cytosine with guanine. Unwinding is followed by complementary strand synthesis. This progresses along the whole length of the DNA molecule. Each contains one strand from the original DNA molecule and one newly synthesized strand that is its complement. That is quite a process. Let us conduct our second activity now, students, and see how much you have learned so far. Working with a partner, please answer each of the following questions.
Students, Let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. Let us check your answers together. Origins tend to be AT-rich, that is, rich in base pairs of adenine and thymine, because AT pairs have 2 hydrogen bonds rather than 3. Therefore, they take less energy to separate. The free nucleotides are assembled into new strands by DNA polymerase. The new DNA strand is complementary to the template because of the base pairing rule, which declares that each nucleotide can only bond with one other type of nucleotide. The ribonuclease removes the DNA fragments that were used to initiate the process of replication. Ligase fills in the nicks, or gaps, that are left in the DNA strands by the ribonuclease's removal of leftover DNA fragments. Did you manage to get the correct answers, students? Great job. You may remember that at the beginning of our explanation of DNA replication, we used the term replication forks. Let us examine this concept in a bit more detail. The replication fork is a structure that forms within the nucleus during DNA replication. It is created by DNA helicases, which break the hydrogen bonds which hold the 2 DNA strands together. The structure then has 2 branching prongs, like those of a fork, each made of a single strand of DNA. These prongs serve as the template for the leading and lagging strands, which will be created as DNA polymerase matches complementary nucleotides to the templates. As you can see from this graph, replication forks involve a large number of elements working in concert. That brings us to the end of today's lesson. We have learned about the mechanisms of DNA replication and the concept of replication forks. Until next lesson, thank you, teacher. Thank you, students.